You're muted.
Okay, I think we can start now. Yeah, so a very warm welcome to everyone here. I am Aishwarya and I'm currently holding the position of Head of Design at Project Encephalon. I am also a physics major and also have a minor in biology at ICET Trivandrum. And today is the third event in the Cajal Week series, where Project Encephalon aims to pay tribute and celebrate the spirit of Spanish neuroscientist Santiago Ramon y Cajal on the occasion of his 169th birth anniversary. It is with special excitement and honor that I'm pleased to welcome today's first speaker, Dr. Nicole Rust. Dr. Nicole Rust is an associate professor of psychology at the University of Pennsylvania. Her research investigates how the brain gives rise to the mind and to behavior with an emphasis on memory. Much of her work lies at the nexus of real and artificial intelligence, as well as experiment and theory. I would like to request the audience to post their questions in the Q&A box, which we shall address in the end. And thank you, Dr. Nicole, for joining us. I would like to hand it over to you now.
Well, thank you so much for having me. It's a real honor and a privilege. I mean, I love what you guys are doing with Project Encephalon. So just the idea of an international trainee-led organization of neuroscience enthusiasts, I just want to applaud it. And you guys have put together such a wonderful event this week. So congratulations, kudos all around. So really excited to talk to you today and share my enthusiasm for something I'm really excited about, which is visual memory. So I will, I'll share my slides. I will say once I begin presenting, I won't be able to see you any longer. I'll only be able to hear you. So if something goes wrong, please interrupt me verbally here. And I'm happy to take questions along the way, however you want to run the show. I'm happy either way. So I'll just begin by sharing my screen here. So can you see me forward the slide?
Yes, we can see it.
Okay, and can you see my cursor move?
Yes.
Okay, great. I will get started. So yeah, so today I'm going to be talking about visual memory. And by that, I mean how we remember the images that we've seen. And I'll be focusing today on a specific question, and that is the question, what makes an image easy to remember as opposed to an image that's easy to forget? So what makes an image memorable? Before I dive into memorability, I just want to talk a little bit about the behavior at large. So this is really what compels me to study this. The fact that visual memory is just so remarkable. So we're really, really good at remembering what we've seen. And this goes back to this classic paper that's aptly titled Learning 10,000 Pictures, published by Lionel Standing in 1973. But the behavior is studied in contemporary labs today, including Ode Oliva's group at MIT. And these experiments, they're pretty straightforward. They typically happen in two phases. So there'll be a first phase in which subjects are asked to just view images and try to remember them. And this will be followed by a memory test. The key of the first phase is that subjects, which will look at images one at a time, each only once, each for a few seconds. The key is that they have to look at thousands and thousands of images, literally, over hours, sometimes over days. So after they've viewed all of these images, which you can think of as a big stack of thousands of photographs, they're given this memory test. So they'll be placed two images side by side, and they'll be asked the question, well, which one of those did you see? So one of these was embedded in the stack of thousands, but they saw each only once, each for a few seconds, the other was not. And remarkably, subjects are really good at this task. So in this Brady study, subjects viewed 2,500 images over the course of 5 hours, and then they were given this memory test, and they were over 90% correct. Now, this was the case when the two images placed side by side were very different from one another. But it was also the case when the images placed side by side were remarkably similar. So here we have two images. In each case, we have bread in a bread box. In one case, the bread is a little bit more in the front. In the other case, it's a little bit more in the back. And subjects didn't do quite as well as if the two images were really distinct, but they did almost as well. So they're 87% correct. Just to be clear, one of these images was embedded in that stack of 2,500, the other was not. They were 87% correct at picking out which one was. So what this is telling us is that we're storing these memories with remarkable visual detail. We're not simply storing memories of the objects that we've encountered, bread, shoe, and so on. We're storing a lot of rich detail about the specific configurations and the context that we see these objects in. So just to summarize the behavior, visual memory is a form of single trial learning. We remember images after seeing them only once and only for a few seconds. It's a type of memory that happens at vast capacity. So we can't find the capacity limit on this type of memory. We just know the more things we see, the more things we remember, and we can remember seeing thousands and thousands of things. It's also a form of a memory that is very visually specific. So we're not just storing low bit information, we're storing a lot of detail. So at this point, we have a pretty good description of the behavior itself. That's not to say that this description is complete, but it's pretty rich at this point. What we really lack is an understanding of how our brains manage to do this. So how is it that we imagine, how do our brains manage to support single trial learning at vast capacity with remarkable visual specificity? And this is where my lab is hoping to contribute. So my lab is hoping to complement all of the really beautiful work that's been done to date, measuring the human behavior, also in human fMRI, by measuring the brain neural activity in the one animal model that to date has been demonstrated an ability to do this, to see images, each only once, each for a few seconds, and report whether they're novel or familiar. And that's the rhesus macaque monkey. And in rhesus monkeys, we can record at exquisite spatial and temporal resolution neural activity. And this is the resolution that we think is absolutely essential in order to figure out how the brain is doing this. And you'll see that throughout my talk. I'll be talking a lot about the nature of the neural code, and the subtext of all of this will be that it's that type of resolution that's required in order to tease apart these different alternative hypotheses and scenarios. So I want to highlight that this type of memory that I'm talking about, it's a little bit different than other types of memory you may have heard of. So for example, one type of memory you might be familiar with is episodic memory. So remember, say, your 5th birthday party or something like that. So this memory is a little bit different. It's called familiarity or recognition memory. And the way I like to explain it is by reminding everyone of an experience I think we've all had, where we're walking down the street and we see someone and we know that we know them, right? So we have that percept of remembering, or that percept that they're familiar. And sometimes this happens in the absence of our ability to recall anything else. So we can't remember who they are or how we know them. We just have this isolated percept of familiarity. So that's what I'm talking about here. That's the form of memory. It's really a binary kind of classification. You can think about it. Think about it. It's the question, have you seen this before? Yes or no? And today I'm going to be focusing on a specific behavioral signature of this type of memory, and that is what we call image memorability. It's simply the fact that some images are easier to remember than others. So while it's the case that generally we're really good at remembering the images that we've seen, we do systematically find some things easier to remember than others. And the question is, well, what determines how memorable an image will be? So just to dive in, here I'm showing you a bunch of images that are configured according to their memorability. So these ones over here are more memorable, and these ones over here are less memorable. And I think if you look at them, you can just see that that's pretty easy to intuit. But I'll be focusing today on a couple of different slices through this question, what determines memorability. In the first part of my talk, I'll focus on the behavior and asking the question, well, what type of content makes images more or less memorable? The second part of my talk will focus on the brain. So what differs in the brain in terms of the activation of a more versus a less memorable image? And then I'll turn to the question, well, what is the origin of image memorability variation? What type of system will have it versus a system that might not? And I want to highlight that all of what I'm going to be talking about today, all of our advances, our ability to answer these questions really follows from techniques that allow us to quantify how memorable individuals' images are. And those techniques were introduced in a couple of papers, both from Aude Oliva's group, the first by Philip Isola, published in 2011, and the second by Wilma Bainbridge, focused on faces, published in 2013. So the way that we score, quantify the memorability of an image is in the context of a recognition memory task. So this simply requires the subject to look at an image and answer the question, have you seen this before, yes or no? So it's a little bit different than the behavior that I showed you in the very first part of my talk. So images are presented in the sequence originally as novel. They'll be replated later as familiar. A subset of these images are often repeated after very short delays, so maybe after one intervening image. We call these vigilance repeats. They're just there to make sure that the subjects are awake and paying attention. They're ones that everybody should get right. But memorability itself is scored after 100 of images or so have passed. So this is over a time scale of minutes. There'll be a repeat of something earlier in the sequence, and this is where image memorability is scored. So each image gets a score based on the performance of the ability of subjects to remember that they saw it earlier. That score ranges from 0 to 1, and it's quite intuitive. So it's simply the fraction of subjects, now this is averaged over about 80 different human individuals, that remember seeing an image. So 1 corresponds to an image that everybody will remember seeing. 0 is an image that people remember seeing at chance, so everybody will have forgotten. And if you look across large data sets, such as the 60,000 images that comprise this data set LaMem, published in this paper here, you see there's quite a range of memorability variation. So there are some images that everybody will remember seeing, or nearly so. There are also images that get scores of 0.4, so less than 0.5, meaning over half of the subjects have forgotten that image. And then there are a range of images in between. The average amount of remembering is 76%. So it's this variation that we're interested in accounting for. So what differs in the images up here that everybody remembers versus the images down here that more than half of all subjects forget? Importantly, these scores are in fact reliable and predictive. So if you measure memorability scores in one group of subjects, and you ask how well do they predict how well another group of subjects will remember those images, the predictions are very high. What this means is that we all tend to find the same images memorable and forgettable. That is not to say that there isn't any individual variation, there is some, but the largest fraction of the variance, the largest component, is driven by shared variation between us. We all tend to find the same images memorable and forgettable. This is not only true across cultures, it's even true across species. So it turns out that monkeys even tend to find the same images memorable and forgettable as we do. And while this is a little bit counterintuitive, really what you'll see throughout my talk is that this image memorability variation seems to be driven by the structure by which our visual system has been constructed. So just how it's been wired up to see and identify objects. This is something that we and monkeys have in common. And I'll be breaking that statement down throughout the talk. Okay. So that's how we score memorability. So we can get memorability scores for individual images, and that's what allows us to line them up on an axis like this, right? These are very memorable images. These are very forgettable images. And the question that I'll begin with is, well, what about the content of the images themselves? What type of content makes an image more as compared to less memorable? Now, the answer to this question is absolutely fascinating. It's not simple. I won't be giving you a simple answer because there isn't one. The first thing to note is that our intuitions about what drives memorability are completely off. They're totally misguided. That is to say, if you take naive subjects that don't know anything about memorability and you just ask them, how memorable do you think this image will be? How memorable do you think this image will be? And you ask them the question specifically, if I show you this, and then I ask you, have you seen it before in a couple minutes, how likely are you to remember this image? So they have a good sense of what memory is. They just don't know about the principles of memorability. That's what I mean by a naive subject. So if you ask them predict how memorable images will be, their predictions are completely uncorrelated with the actual memorability scores, which again are predictable and reliable. So for example, naive subjects will predict that this image is very memorable. It's this beautiful picture of this waterfall, but it actually has a pretty low memorability score. Only 58% of all subjects will remember seeing that image. In contrast, subjects will predict that this unremarkable kind of ugly bathroom scene is very forgettable, and yet it turns out that this bathroom scene is pretty memorable. 85% of all subjects will remember seeing it. And if you plot for individual images how memorable people think images will be versus how memorable they actually are, you can see that there's basically no correlation here. So what's going on? Well, it turns out that people's predictions about memorability are driven by how interesting or aesthetic, how beautiful the images are. And this makes sense if you look at these images. So this waterfall is quite beautiful, right? And so that's why people are predicting, well, I'll remember that, it's absolutely beautiful. This bathroom, on the other hand, it's pretty ugly, right? Not very beautiful. I'm not gonna remember it. So people are basing their predictions on how interesting or beautiful images are. And we know that because we can also ask people, well, how beautiful is this? And how interesting is this? And those predictions of beauty and interestingness, those are highly correlated with memorability, with people's predictions of memorability. Sorry, let me say that again. The people's judgments of how interesting and aesthetic images are highly correlated with how memorable they predict images are. But those predictions are not correlated with the actual memorability scores. Okay, so what we think memorability is turns out not to be what it actually is. But because we can quantify memorability, we can look to see, well, what is it? And a few principles have in fact emerged. So we know that images with people tend to be quite memorable. In contrast, images of nature scenes tend to be quite forgettable. It's also the case that atypicality tends to be quite memorable. So if you see a typical image in an atypical situation, that will be more memorable than if you see a typical version of that same image. That's also pretty intuitive. There are a lot of different types of factors that drive memorability. So things like emotional valence. Images are more memorable if they evoke disgust or amusement, but they're less memorable if they tend to evoke awe or contentment. There are a large number of other types of factors, including some low-level properties like the brightness or color or the size of an object in an image. It's also more memorable if it has an uncluttered background like this, as opposed to embedded in a highly cluttered scene. In the case of faces, we find them more memorable if they appear intelligent or responsible or trustworthy or kind, and so on. So you can see a large number of different types of factors, low-level image properties like brightness, emotional valence, face interpretation of personality based on facial characteristics. All of these things combine to determine memorability. The answer to the question, what types of content make images more versus less memorable? The answer to that question is it's really, really complicated. So just to summarize what I've told you thus far, we know that the mapping of image content to image memorability, we know that it's counterintuitive, and we know it's highly complex and multifaceted. At the same time that might seem all a little bit hopeless, we also know that this mapping is highly predictable. And we know that based on new developments in machine learning. So a computer vision model exists, it's called MemNet. And what MemNet can do is take any image and produce a prediction of how memorable that image will be. And its prediction is very, very good. So what this computer model is a deep convolutional neural network. So this is a machine learning algorithm. Sometimes this is called artificial intelligence. So these are deep convolutional neural networks. And it was trained to take the images that comprise this data set LaMem, again, 60,000 images and their memorability scores. And this computer model was trained to do what it does, predict how memorable images will be. And its predictions are very good. So it captures 95% of the variance that's explainable across humans. So it's 95% good at predicting image memorability. What this tells us is that the memorability of images are in fact predictable based on just the pixel patterns, the images themselves. Okay, so it's not a mysterious, unmappable quantity. It's something that in fact can be determined. So just to build on what I told you before, it is the case that the mapping of image content to memorability is counterintuitive and complex, it's multifaceted, but it's also predictable. All right, So that's the behavior. It's kind of complicated, but it's not mysterious or ineffable or ununderstandable. And so now we'll turn to the brain, where counterintuitively, if we look in the brain, the explanation, at least to date, seems a lot simpler. So here we'll ask the question, well, if we look in the activation in the brain for a more memorable versus a less memorable image, how do those differ? And the project I'm about to tell you about was a fun collaborative effort between my group and the group of Aude Oliva at MIT. And in the lead, we had this fantastic set of postdocs, so Vahid Mirpour, Drew Cajal, and Travis Meyer, all from my group, and also Yalda Mosinzada, who was working with Aude at the time. And we began by knowing where we should look in the brain in order to account for image memorability variation, and that is in high-level visual cortex. And this was based on some really nice work from Wilma Bainbridge in human fMRI, where she determined that you can decode whether images are more memorable or less memorable within a class of, say, faces or houses by looking at the activation in high-level visual cortex. So I will call this the human analog of what we call infratemporal cortex IT, which in the monkey is here. So IT is a brain area that's been implicated extensively in supporting our ability to identify objects. So to figure out what objects are out there in the world that we're looking at right now, whether they're faces or coffee cups or pencils, and especially to solve that problem despite all the variation with which those objects can appear. So the problem is computationally quite complex because faces and coffee cups and pencils can exist at different positions and sizes and in different backgrounds and different poses. And so our brain has to figure all of that out. Is that a face or is that a pencil and so on? And computations in IT cortex are known to be contributing in an important way to supporting our ability to recognize objects. So IT is considered to be the highest stage in the what we call the form processing pathway in the primate brain. Form processing meaning it helps you figure out what it is you're looking at. So signals come from the eye and then they travel into the thalamus and then they go through V1, V2, V4, and then to IT. After signals leave IT, the signals become multimodal. So they go into the medial temporal lobe, places like perirhinal cortex, and they also go into prefrontal cortex. So that's why we think of it as the highest stage of purely visual processing. So IT has been implicated extensively in supporting our ability to recognize objects. The results of human fMRI suggest it also has some sort of representation that drives whether or not we're going to remember and see an image, or at least drives this image memorability variation. And so what I'll be addressing is the question, well, how is image memorability reflected in IT? And how is this integrated with these object representations that we already know to exist there? So I'll just start with the first question. So how is memorability reflected in IT? And I'll just summarize by jumping to the punchline of what we found. So the experiments we performed were experiments in which monkeys were performing a task that's very similar to the one that was used to score memorability in humans. So on each trial, the monkeys look at a single image and they make a behavioral report about whether that image is novel or familiar by moving their eyes to one of two targets. So in this case, an upward saccade to this target means the monkey thinks it's a novel image, and downward means it's familiar. So the images are presented earlier in the sequence as novel, they're repeated later as familiar. And as the monkeys are performing this task, we're recording neural activity in IT cortex. So we're recording extracellular activity. Our data consists of hundreds of individual neurons and their responses to hundreds of images. And for this analysis, I'll be focusing just on the novel, the first presentation of an image. So the question here is, what differs in IT cortex in the first time you see an image when it is highly memorable versus when it's more forgettable? And the answer is the vigor of the firing rate response. So this is a plot of how memorable individual images are as a function of the vigor of the response in IT cortex. So technically, this is what's called the L2 norm. It's the technically correct way to measure the population response magnitude. each image here is each dot is a single image. And you can see there's a strong and highly significant correlation between how memorable humans find these images and the vigor of the response in monkey IT cortex. It's also the case, and I'm not showing you that, but it's in the paper, that monkeys and humans tend to find the same images memorable. So this is also true of monkeys too. Now this axis is a little non-intuitive, so I'll just transform two of these points on this plot into firing rate responses, which if you're familiar with firing rates, will probably be more intuitive to you. So a highly memorable image evokes a grand mean firing rate in IT cortex of 15.5 spikes per second. And that drops down to 12.5 spikes per second if that image is less memorable. So this is about a 20% reduction in the overall vigor of the response with changes in memorability. Okay, so this result is consistent with the general interpretation that what's driving memorability is the fact that you have more robust visual representations. So more robust visual representations are then stored as more robust memories. So that's what this result suggests. It's a type of coding scheme, it turns out, we did not know before this existed in IT cortex. So now I just want to explain to you, when I start talking about coding schemes, how we think this coding scheme is what we call multiplexed or integrated with other things that we know IT cortex is doing. So I'm just going to give a little primer to start out with what others have determined about IT cortex, and then about its object-based representations. And then I'll extend those to account for memorability as well. So if we think about IT cortex and how it reflects or represents information about the identity of what you're looking at, we know that if you look at an image, this causes a wave of activation that ends up here in IT cortex, and it is encoded as what I like to call a pattern of spikes coding scheme. This simply refers to the fact that some of these units will be activated by the fact that you're looking at a flower and others won't. And so you'll get a particular pattern every time you look at a flower. Now, we like to think about these patterns of spikes coding schemes in what's called a geometric representational space. It looks like this. It looks a little complicated, but it really isn't. It's simply the plot of the vigor of the response of the first neuron versus the second neuron, and so on. Technically, there are 16 million neurons in IT cortex, and so technically this is actually a 16 million dimensional space. But we can gain all of our intuitions in just this few number of neurons, like the ones I've shown here. So this will drive our intuitions of what's happening. And so it turns out if you look at a flower, that's going to cause a particular pattern of spikes in IT cortex. And that's going to end up, it's going to be reflected as a vector somewhere in this high dimensional representational space. Again, it's just the plot of the vigor of the vigor of the response of neuron 1 versus neuron 2 versus 3. That pattern causes a vector. This vector is going to position here when you look at a flower. Now, if you change what you're looking at and say you start looking at an apple, that's going to cause a different pattern simply because some of your IT neurons are more responsive to apples than they are to flowers. So that's going to cause a vector that's pointed in a different position in this high dimensional representational space. And one of the things we know about IT and what makes it special is that the representations of images with similar content, like all these pictures of flowers, they cluster together in the IT space, meaning that the brain can look down and look at the representation here and say, oh, I'm looking at a flower because the response is over in this region of the space. It has that sort of, it has the flower pattern. That's the general idea. In other words, changes in the direction that these vectors point are what we believe, and we have good evidence to suggest that changes in the direction that these vectors point reflect changes in the identity of the objects out there in the world. Now, that's object recognition. Now we can ask the question, well, how does IT simultaneously support representations of object identity as well as memorability? And there we find that memorability is reflected in a complementary coding scheme. It's variation in the length or the vigor of the responses of the overall population response. So we know that images that are more memorable evoke more vigorous response, which correspond to longer population response vectors, where images that are less memorable evoke less vigorous response. And so this explains how IT can simultaneously cluster information about object identity and its population response patterns, while also supporting this representation of image memorability by not changing the pattern of the response, by rescaling it to be more or less vigorous. That's what these results suggest. So, brain. What differs in the representations of more memorable images? More memorable images evoke more vigorous binary responses in IT cortex. It's a coding scheme that we did not know existed there, but it's one that's strongly correlated with this behavioral phenomenon, memorability. Now we'll turn to the question of the origin. Well, where is this memorability variation coming from? So when I say origin in this case, I'm referring to the question of, does this memorability variation follow from any system that's optimized simply to see, or does that system also have to be optimized to remember what it's seen? And we can ask this question because of the existence of a class of computer vision models that are wired up to see, but they're not optimized for memory and they can't remember anything. And so here I'm referring to deep convolutional neural networks and something that's really interesting that's happened in this field over the past, I would say a little less than a decade or so. So the observation, which is quite compelling, is that if you wire up one of these computer vision models to recognize objects, and so why would you do that? Well, Google does that, for example, if they want to tag particular images. So know that this image has a dog and this image has a flower, such that when you search for dog in Google, all of the images with dogs will come up. There are a lot of different reasons that computer vision would want to be able to solve the problem of object recognition. And so there's been a big industry in computer vision of trying to build models that do just that. So there was a big breakthrough some years ago on how you can train these models to do that. And there was an observation that followed, and that is when you train a deep convolutional neural network to recognize the content of images, so the objects that are present in images, what you find is a model that has an organization, an activity pattern that looks a lot like a brain. So these models have different layers to them. So the early layers have activity patterns that look like early visual cortex, so places like V1. And as you go deeper into the layers, they map onto deeper stages of processing in the brain. So V2, V4, and then the highest layers, IT cortex. And so this is an observation that was made by individuals like Dan Yammons and Jim DiCarlo and Nico Kriegeskorte. These models go by the names of AlexNet and VGG16. You may have heard of those, and so I mentioned them in case it helps you connect the dots. So these models have been wired up just to see. So they're just wired up to see. Once you train them to do that and then you fix their weights, they can't remember anything. Meaning if you show them an image the first time or you show it to them the millionth time, they'll respond in the exact same way. That's what they're trained to do, and that's all they can do is just identify the content of images. So we asked the question, well, do these computer models, do they also have a representation of memorability? So is it the case that, like the brain, if you go to the IT-like layer of these networks, will they respond more vigorously to some images than others? And is it the case that the images that they respond the most vigorously to are the ones that we remember best? Just to show you those results, I'm going to summarize what's happening in the brain with this R. It's just the Pearson correlation between the memorability scores for individual images and the vigor of the response, right? So we have this strong correlation. And now we can plot that correlation across different layers of the network. Here I'm showing you the results for VGG16, but these results are similar for every one of these networks that we've tested. What we find is that in the earliest layers of these networks, the correlations between population response magnitude and image memorability, these R values, are small, and they're no stronger in the trained version of the network than if you just randomly connect a network together before you train it. But as you ascend to the IT-like layers, what you find is a network where you have correlations between image memorability and network activity. Meaning that at the higher stages of these network, at this layer here, right, in this computer model, again, just wired up to C, it is the case that this layer will respond more vigorously to some images, and those images are the ones that we humans remember best. Because these networks are not explicitly trained in any way to represent image memorability, what this suggests is that memorability is just emerging from these networks as a computations of the optimizations required to wire up a system to C. So that's the origin story. That suggests that this memorability variation at the end of the day is really just a consequence of vision. It's not a consequence, or at least a component of it is a consequence of vision, as opposed to memory per se. So I'm just gonna take a moment to summarize what I've shown you so far. Okay, so we've asked the question, what determines how memorable an image will be? And you can ask that question from a number of different perspectives, and I took 3 of them. The first perspective we took is the behavioral perspective. And we asked, well, what type of content makes images more versus less memorable? Well, I told you some principles, like faces are more memorable than nature scenes, and atypicality is more memorable than typicality. I also told you that the mapping of image content to memorability, it's very counterintuitive, and it's very complex and multifaceted, and there's no simple explanation, simple way to describe it. But we do know that it's predictable because we have computer vision models that can predict with really high accuracy how memorable images will be. We then took the perspective of the brain. So we asked the question, well, what differs in the brain in terms of the representation of a more as compared to less memorable image? And there I demonstrated that it's reflected by the overall vigor of the population response in high-level visual cortex. And then we asked the question, well, where does that come from? And we found that it emerges naturally from computer vision models that are just wired up to see, at least a component of it does. So together, all of this kind of points to the fact that memorability is really a consequence of vision, right? So a more memorable image for all sorts of complicated reasons that are hard to describe back in the content space. More memorable images evoke more vigorous visual responses, and that is what drives memorability behavior. Okay. So that's one component of the story. But it's not the whole story, because it also is the case that not only does the brain have to have this activation by the incoming visual input, but it also has to store it in memory. So there's a second part of this story as well. And so I'm going to just end my talk by talking a little bit about memory. So the conversion of a visual representation into a visual memory. Okay. So what do we know about how we remember the images we're looking at? Well, we know at least where we should be looking. And so that is in the medial temporal lobe complex, these memory structures you've probably heard a bit about before. So places like perirhinal cortex, entorhinal cortex, and the hippocampus. And in terms of their visual input, they receive input from infratemporal cortex IT, the structure that I've been talking about. Now we know the structure perirhinal cortex in particular is really important for our ability to remember images. In fact, it's thought to support the vast capacity nature of this, the fact that we can remember thousands of images after seeing each only once. And we know that lesions to perirhinal cortex produce profound deficits in the ability to report whether an image is novel or familiar. At the same time, we also know that it's not the only structure involved. So it's also the case that the hippocampus is contributing, but it's contributing in a really interesting way. So it's thought to shape the visual specificity of visual memory, our ability to remember whether we saw the bread in the front as compared to the back of the bread box. And evidence for this comes from lesions in human patients, either by stroke or by because they've had a surgery to correct epilepsy. So patients with hippocampal lesions, they do something interesting. They overgeneralize their memories. So disproportionately relative to the rest of us, if they see the bread in the back of the bread box, if you ask them if they saw it in the front, they will disproportionately falsely remember that they did. Okay. So this suggests that the hippocampus is shaping the visual specificity of visual memory. So it's not just a single structure in the medial temporal lobe that's contributing to this behavior. It's an interaction between multiple structures. It's also the case that we don't think about this pathway as purely feed forward. So in general, we think about memories are probably stored to some extent somewhere in the medial temporal lobe, but there's evidence to suggest that they are then fed back to IT cortex. And evidence for this includes, again, a case study of a patient with a hippocampal lesion that had blunted memory signals back in visual cortex itself. Okay, so just to summarize, what we know suggests that there's a whole lot going on here. There are multiple structures contributing. It's not just feed forward information, but we also have information that's going forward and coming back and so on. And so it's a really complex pathway, and as is the case for the non-human primate as well as our own brains. These are really complex systems in order to pick apart. So the way that my lab has been approaching trying to figure out how is it that our brains support our ability to remember images is to begin by trying to pinpoint the so-called neural signal that drives the perceptive remembering. So insofar as you're walking down the street and you have that aha moment, you have that moment where you remember that you've seen somebody before, there must be some sort of activity in your brain that's driving that percept. And the question is, where is that activity located and how does it manifest? Once you pinpoint the so-called neural signal driving memory, then you can ask, how does it come to be? And that is the answer to how do we remember images? In the absence of understanding where that signal is, it's really hard to do anything else. This is the logic behind our approach. So we've been very focused over several years on this first step, pinpointing the neural signal. So I'm just going to summarize very quickly the history behind where the field was and what we've contributed over the past handful of years to our understanding of this neural signal. Okay, so there's been a hypothesis around for some time, going back to the early 90s, that the signal that drives our perceptive remembering is a phenomenon known as repetition suppression. So repetition suppression, its existence is not in dispute. It's just an observation of a phenomenon that exists in IT as well as throughout the medial temporal lobes, so parietal cortex and the hippocampus. It's the observation that when you look at a novel image, you get a vigorous response out of these brain areas. And if you look at it again, you get a decrement in the response. So that's repetition suppression. The question on the table is this phenomenon the signal that drives our perceptive remembering, or in the case of a monkey's behavior? So there are a lot of reasons to think that it might. So like the behavior, repetition suppression is highly visually specific, meaning it has the capacity potentially to support memories for thousands and thousands of images. So if you get repetition suppression for, say, a flower, so you see a flower, you'll get repetition suppression for the flower, but that won't transfer to another object like an apple. You'll only see repetition suppression for the apple if you actually look at it a second time. It's also the case that this signal, this phenomenology, happens after you see an image only once, and it lasts an incredibly long time. So according to Zhang and Brown, it lasts up to 24 hours after seeing an image. In more recent work, we demonstrated that the rate of its existence and disappearance is well matched to the rate of remembering and forgetting behavior with time. These are quantitatively well aligned. And one could imagine with this so-called signal, how the brain could interpret whether or not something is novel or familiar. So it could do something, in the simplest case of just counting the total numbers of spikes coming out of IT. So if the total numbers of spikes, if the IT is responding vigorously, the brain would interpret that image as novel. And if it's less vigorously, the brain would interpret that image as familiar. What I'm describing to you can be mathematically instantiated as a class of what we call linear decoders. And it's an entire family of linear decoding schemes, again, that we've determined, at least in some cases, provide a nice mapping of quantitatively of IT neural signals onto behavior in terms of time of remembering and forgetting. Okay, so that's some history in the field. It turns out, however, there's a big problem with this hypothesis, and that is what I'll call so-called elephant in the room. That is the fact that factors other than memory, we know, modulate the total numbers of spikes or the overall vigor of the firing rate response in IT cortex. So if you make an image brighter, IT cortex will respond more vigorously. If you increase the contrast of an image, IT cortex will respond more vigorously. It's also the case that the memorability variation that I talked about in the entire first part of my talk, that also, it turns out that goes in the wrong direction in order for this hypothesis to account for the behavior. In the simplest case, this hypothesis predicts that because these stimulus-associated factors will change the vigor of the response, the hypothesis predicts that we will confuse changes in the brightness of an image with whether or not we perceive it as novel or familiar. We don't make those confusions. And so the inference is that the hypothesis cannot be correct. So the question is, well, if that's not the memory signal, then what's the alternative? Okay. So in very recent work, in fact, in a paper that's published today, in PNAS, we hypothesized that repetition suppression is largely on the right track, but it needs an embellishment. And that embellishment we call sensory reference suppression. This is the hypothesis that what the brain is doing is estimating the level of activation expected from a sensory input and correcting for it, leaving behind the signal for familiarity. And so I'm not going to do a deep dive into this study because it's beyond the time that we have together today. But I'll just tell you that our general approach for this was to test the ability of this sensory reference suppression, which you can technically instantiate as a linear decoder from IT cortex, or let me just say you can interpret this signal. So we tested the ability to interpret this signal coming out of IT as in terms of its ability to account for the rate at which monkeys could disambiguate changes in familiarity from changes in contrast. So we trained the monkeys to report that if they had seen an image, even if it changed its contrast, it's the same image, they should still call it familiar. Humans can do this, monkeys can do this robustly as well. They don't confuse changes in familiarity and changes in contrast. And it turns out that SRS, if you decode that from IT cortex, that provides a much better account than does this repetition suppression. So together, all of the evidence is pointing to the idea that it's not quite repetition suppression, but there is a signal that's a lot like that. It's called sensory reference suppression, and this is the signal that's driving the percept of familiarity or the percept of remembering that you've seen something before. Okay. So now we feel like we're in a good position. We have a good candidate for the neural signal that is driving this familiarity behavior. And so now we're moving on to this step, trying to figure out where does sensory reference suppression come from. And so in our current work, we're recording not only from IT cortex, but also from the medial temporal lobe to tease apart the system and understand how is it that we remember the images that we've seen. So stay tuned for future work. All right, so I'll just end there. I'll end with a big thank you. I want to wish a big happy birthday to Cajal on this fine day, and then the celebration of him with you. And so to learn more, I've highlighted some of these papers summarized here, and I'm very grateful to our funding agencies who have supported this work. And yeah, happy to take any questions you have in our last few minutes together.
Thank you so much, Dr. Nicole. That was a very insightful talk. and clearly very interesting. Okay, we can take questions now. So, um, uh, Sweekrut, would you like to come live and ask your question? Yeah, so he'll come live.
Okay, am I audible? Am I visible? Am I around? Okay,
I hear
visible.
you. I hear you. I don't see you, but I hear you. So that's good.
Cool. So first of all, thanks a lot, Dr. Rust. That was a really eye-opening talk. And coming to my question, so this has got more to do actually with my concern for people who have PTSD and might have triggers, like especially corresponding to certain images which they might associate with something bad that happened in their lives. So
yeah,
the emotions do play a rather important role in memory, right? So
yes.
is there a particular how do I put it? It seems maladaptive in some cases. Actually, that it would be worth forgetting certain images compared to certain other ones. But has this been tested in people? Has it been seen that what role does emotion play, and whether some people have a harder time forgetting certain memories compared to others?
Yeah, no, this is a really fascinating space. It's also something that fascinates me. It's not a space that I work in, but it's a space that I'm sort of aware of just because I'm so fascinated by it. So there's another structure that I haven't talked about at all. It's called the amygdala. And the amygdala is thought to play a really important role in emotional memory. And yeah, there's a lot going on in the space of PTSD and how do you treat it? So that, like you said, there are these really salient triggers that cause, tremendous trauma. So people will re-experience these traumatic events that they've experienced in the past. And the question is, how can you treat these people? And so there are fascinating attempts at this. I think, there has been some success under very controlled conditions. If people re-experience the event again and again, they can not necessarily forget it, but they can get some emotional distance from the event. But even the re-experiencing, it can be quite traumatic. And so there's some fascinating things going on. Both chemically, I know that there are a lot of people working on how can you modify memory, is there any sort of drug that can help forget, or that you can administer and have online when people are remembering this so they'll never remember it again. There's also this fascinating fMRI approach, which is a biofeedback approach where people are trying to activate a memory kind of sub-threshold so they don't perceive it, but yet they can activate it enough to extinguish it. So there's really some fascinating work, I think, going on, but it's all very much in development right now. And we don't, as far as I know, we have treatments for PTSD, but they're still suboptimal. And so it's definitely a space to keep your eye on. Yeah.
Really fascinating. Thank you very much.
Okay,
and I guess I shouldn't say just keep your eye on, contribute to. You guys are a bunch of trainees who are, neuroscience enthusiasts. I mean, yeah, what a wonderful way to, what a wonderful career trajectory to go into, right? That's such a wonderful, I think, that's both in the short term and the long term. Yeah.
Okay. The next question we have is from Amartya Pradhan. He asks, did you check what happens to the neuronal firing frequency from IT projections to the hippocampus, if any, which is highly implicated in memory?
Yeah, so we are currently working on that. So literally right now, I think there are probably electrodes in hippocampus as monkeys are performing memory tasks, right? So literally right now. But yes, we're in the middle of a study looking at what's going on in the hippocampus and some other structures. It's too early to really comment on what's going on there at this point. I mean, I'll start with the observation that in human patients with hippocampal lesion, it's remarkable that if you just ask them to do the Lionel standing task, right? So they look at images and have you seen this before? As long as you're not asking them to discriminate between images that are visually similar, they're unimpaired in that task. So people can remember images that they've seen really, really well, even if they don't have a hippocampus. It's absolutely remarkable. So that suggests that the memories for visually distinct images are stored somewhere else, and that the hippocampus comes online when you're trying to remember whether you've seen something similar to something you've seen before. That's the, you know, the current story right now in the literature. Yeah, and so I'll keep you posted as to whether or not that story seems to hold up or not. But that's what we know right now. Yeah.
Pratibha, would you like to come live and ask your question? Pratibha? Is she here?
Hello.
Our picture pop up.
Hello, am I audible?
Hello? Yeah, we can hear you.
Yeah, so that was a mental problem. Does this image memorability depend on the emotions, like either the emotions which are related to an image or any emotion which is shown in the image? So does the emotion in the image actually affect the memorability of the image?
I'm sorry, I couldn't hear that question. There was a lot of background noise. Did anyone else hear it? Can they give me a summary?
I'll read it out for you. So basically, she wants to ask, does this image memorability depend on the emotions either related to the image or portrayed by the image?
Yeah, so it's definitely the case that for humans, we know that images that evoke certain emotions tend to be more memorable, and images that evoke other emotions will tend to be less memorable. And I had that on a slide a little bit earlier. If you're interested in digging deeper in that, there are a couple places I would point you to look. One of those is we did a review of memorability in Trans and Cognitive Sciences, but the original article is the Isola paper that I referenced throughout my talk. So yeah, we know that there's a correlation, at least, between how much certain emotions evoke. When you look at an image, it evokes a certain amount of emotion. You can ask people to score how much awe do you feel, how much contentment do you feel, how much disgust do you feel, and that those factors that people score will be correlated with how memorable images are. But we know almost nothing about how that is actually happening in the brain. We know about the behavior, but not the brain at this point. And so I think, for example, one big question is that emotional component. Are you going to see it reflected in, say, high-level visual cortex? Or is that something, and maybe that happens because the amygdala feeds into high-level visual cortex, or maybe that's something that isn't reflected in high-level visual cortex at all, and you have to go downstream from visual cortex, and maybe that's when the amygdala starts to contribute. We don't know the answers to those questions yet. And I don't know that the monkey is the best model in order to figure that out, because I think it's really hard to figure out certainly some of these factors like awe and contentment. I don't know whether monkeys are capable of awe and under what conditions they experience it. But I think that's our human fMRI would be the right way to approach that as a first pass, for sure.
Next, we have Bhishmaraj Selvamani for asking the next question on live.
Am I audible?
Yep.
Hello.
Yes.
It was really nice talk and fascinating.
Yeah,
I had a couple of questions. So one was based on like a few examples you gave. It seems like there's a strong evolutionary angle to it, where images which are like useful in some sense are more memorable. So I thought, like, was that also considered, like, as to giving an explanation as to why certain images are more memorable?
So you're asking about like the evolutionary angle. I mean,
Yeah.
there's a sense definitely in the, I don't know, I guess when you say useful, you have to say useful for what. And
Yeah,
certainly
I mean,
the deep neural network results speak to the idea that, I mean, the network is trained to do one thing, and that is recognize the objects out in the world. And so whatever computations are useful for that, image memorability kind of emerges naturally from, you know, from that. So certainly I think that there's a strong evolutionary component as long as the end goal is recognizing objects. We don't have any evidence for anything beyond that at this point, but that's not to say that there isn't something beyond that. We just, that's all we have.
Okay. Yeah, the other thing was, so it seems like when you showed the examples, like images which were like novel or in some sense more informative, like so like very low chance of happening, are like more memorable. So yeah, it just was like interesting to see that play out, like where it's like images which are like common are in some sense like less memorable, right?
Yeah, the atypicality aspects to it. Yeah, I agree. That's really interesting. And I was kind of surprised, I mean, about the fact that you have to, I guess, get a little bit deeper into the deep convolutional neural network kind of world in order to have any intuitions for how it works. But the fact that a computer vision model would also respond more vigorously to something that's atypical kind of surprised me, because intuitively I would think, well, if the network is trained to do something, you think it would respond best to the thing it's trained to do. And yet that's not what these networks do. They respond more vigorously to things that are unusual, is the inference. So yeah, so it's kind of surprising finding and non-intuitive.
Another small thing, like I just have one thing with the task. So does that like the images shown later are more memorable? Like was that considered like in some sense, because maybe recently shown images are easier to recognize than the images shown earlier, because there are like 60,000 images, right?
So it's not the case that in these experiments, there's constantly a mixture of novel and familiar images shown. So it's okay. Nothing to do with recency. It's always the case that these images are always shown with the same delay. So you can show something and show something else 100 images later, and then you'll show something new here and 100 images later, and so on. It's also the case that different subjects see these images in different orders. And so, yeah, there's really, Ode and her team have been very careful about dissecting and making sure that it isn't an artifact of any of this other stuff. It really is about the images per se. Yeah.
Sure. Yeah. Thanks. Yeah, that's it.
Okay, next we have a question by Deepak. He asks, is it right to say that these deep neural network models are merely a bunch of super specialized filters with a bit of memory to infer what's in the image if trained properly? If that's correct, how close do you think are we to create a ultimate deep neural network model with super generalized filter with a bunch of memory to infer and learn on the go like a human baby? Thanks for the great talk. Yeah.
Yeah, no, I mean, this is a fascinating question. I hesitate to put adjectives like, are the deep neural networks just? It is the case that the deep neural networks, the components that they're made of are quite simple, right? There's no magic in the box. They're made of a bunch of very simple elements. And what comes out of them is a behavior that's quite complicated. And before somebody observed that you can train one of these things up in order to recognize objects and it looks like a brain, we really didn't have a good model of how object recognition worked. We had some sketches, but we didn't have a model that could predict how IT cortex would respond to arbitrary stimuli. So So yes, they're made of simple elements. Yes, those simple elements together produce something really exciting. But I think there's been a lot of really fantastic work going on in the field and a lot of conversations about, well, what are the limits of these types of approaches? And for this work, I would point to people like Josh Tenenbaum at MIT and his work on intuitive physics and his demonstrations that there are some things that human babies do that these deep networks seem to have a really, really hard time with. And that those are kind of the definitions of human intelligence and the magic in the box, right? And that those are the things that are kind of by definition interesting. So you can ask about human babies and what they can do and what deep nets can and can't do. You could also ask about how does a human baby wire up itself to just recognize objects? Like that's a different question. So unquestionably, these deep nets can categorize objects to some extent. In fact, I think at this point they're doing it better than humans, although they don't do it like humans. So they can do that. And then the question is, well, how does a human baby learn to do that? And is that mimicked in how we train these deep networks up? And the answer there is unquestionably no. Babies are not given millions of labeled examples in order to wire their systems up to see. And so that's where this field of unsupervised learning has really taken off. And there's some beautiful work going on there. Yeah, so a very multifaceted and nuanced answer to your question.
Next, we have Arun. You may please ask your question.
Yeah, Dr. Rust, can you hear me?
Yep.
Yeah, so I will go on with my question. So actually, when you talk about the images and memorability, you talk about the temporary memory, as I guess. Like
you talk about what kind of memory?
Like a temporary memory?
A temporary memory? Oh.
Yeah. Yeah, for example, if you show me image of a person, maybe I would remember it for a few hours, or if you show me image of a place, I would remember it maybe later on if I
so it definitely the images,
something like that.
Yeah, the memories degrade with time, but they're also it's certainly the case that I could show you images now and I could come back in 6 months and you will remember some of what I showed you. Yeah, so it's not super temporary.
So if like that, like if you show me some image after 6 months, what is the probability of you remembering an image? Like, will it be same like if you show me in the earlier phase or in the later phase?
So it definitely degrades with time, but it can be quite robust. I mean, I don't all of these things depend on the specific conditions, and I can't rattle off exact statistics to you.
Yeah.
But for example, when I mean conditions, I mean how well you remember something depends on how long you've looked at it. So you will remember something better that you looked at for 2 seconds than something you looked at for 1 second, for example. So I can do a lot of little things to manipulate how well you'll remember. But the question you're asking is whatever rate of remembering you have right now, how does it degrade as a function of time? And I know some of the principles along which that is true. So we know that these image memorability, their ranks, they're maintained across time. So you forget everything such that memorability ranks are generally maintained across a day, a week, and a month. It's also the case you will have residual memory across a month. You will still remember the most memorable images will still be the most memorable to you. The least memorable images will still be the least memorable to you. But your overall rate of remembering will be worse at a 1 month time point than it was at a day or a week. But yeah, the one thing I can't answer because I don't have one off the top of my head is what would that rate be? But I could certainly point you to the papers. If you send me an email, I can point you in the right direction, or you'll see it in the TICS paper as well. We cite that literature.
Sure. Thank you for your answer, and congratulations for your paper.
Oh, thanks.
Okay, I think that's all for the questions. We are a little bit ahead of our time. Thank you so much for answering all the questions very patiently, Doctor. And I would like to hand it over to Pranjal now.
Well, thank you so much. It was really such a pleasure to join you all. And again, I love what you're doing. I love to know about Project Encephalon. So, I just want to cheer you guys on, keep it going, and keep in touch. So thanks.
Thank you so much. Pranjal, I think you are on mute.
Maybe now?
Yeah.
Hi, I am Pranjal, current president of Project Encephalon. Thank you, Dr. Nicole, for a very exciting talk. At the end of each talk, I tend to acknowledge some members of my team without whom organizing Cajal Week would have been impossible. Today, I'm acknowledging the work of Pratiksha, Sheetal, and Payal for making the exhibition from Cajal Legacy possible. As you can see in the opening presentation, there were different images. So those were from Cajal Legacy. Next talk will start approximately in 40 minutes. The link for the talk can be found on a social media web page, and I will also be posting the link over here. And in fact, Amarth has already done that. Thank you. Thank you, everyone. See you in 40 minutes in Dr. Sean's talk. Thank you so much. Thank you, Dr. Nicole, for joining.
Bye, everyone. Thanks so much. Great to meet you.